Okay, today we're going to go over the ecological basis of succession. So we've looked at basic insects and things that we expect to see on dead bodies. Now we're going to see what order we expect them to show up in. There are a lot of methods for determining postmortem interval. These things include histological methods, things like the aging of the tissues as they decompose, chemical methods, different chemistry changes in the eye, other areas of the body, bacteriological methods, so the bacterial succession on the body, and zoological methods, so the insects on the body. So the zoological method, this is where we come in. The zoological way is a way of determining postmortem interval based on the animals associated with carrion. So the word faunistic, when we look at faunistic studies, refers to fauna, or all animals occurring in a specific region or during a specific point in time. So the zoological method of postmortem interval estimation is focused on the faunal succession on carrion. The basic ecological premise behind this idea is that each wave of organisms using a resource is unique and it changes as that resource changes during exploitation. So as these insects or as these different animals feed on a body, they change that body. So if you think about it, there are some of you that I've noticed have been watching different animals decompose or different things attract flies out in the wild, or you've seen this before. When you first come upon an animal, you'll notice that that animal's fresh. It isn't really stinky. Nothing like that is happening. Then once flies show up, they begin to feed on the body and it accelerates the decomposition. So that first wave of feeding insects accelerate decomposition. They change that body through removal of tissue, through excretion of waste products, all that sort of stuff. They'll end up changing it so much that that same species of insect is no longer attracted to that body. Okay? Then they change that. They've changed it so much so then the next group of insects that come in are attracted to the body in its new form, and they change it. And then the next group is attracted to the body in a new form. So if you remember back when I had you watch the entomologists at the Casey Anthony trial, I believe it was Dr. Tim. Why can't I think of his last name? It was Tim, who said that insects like bodies much like people like their steak. Some people like it really, really rare. Other people like it well done. Insects the exact same way. Some like it fresh. Some like it decomposed. Some like it dry. Okay. So the succession of insect waves will change the resource during exploitation. Now the most common carrion or most common fauna found on carrion is insects. Yay! Hence the whole forensic entomology thing. And insects can accurately estimate the age of a corpse. So that's nice. We've tested this many times. It's been looked at over the years. So we're good to go. Now there are some limitations, however, to the use of faunistic studies in corpse age estimation. So this is something that you have to keep in mind whenever you use any forensic technique. Doesn't matter what it is. You have to keep in mind those limitations. We've been over a few of these limitations in the past few weeks, but let's look at some of the major ones. One, the first major limitation for faunistic studies is that the estimation is based or dependent on accurate data. If no one has accurately looked at what organisms show up when on a particular body, then accurate PMI estimation is difficult. So if we don't know what's supposed to be there, we don't know when they show up, we don't know how long they live, we have a really hard time estimating how long that body has been there. So it's dependent on accurate data. Two, it depends on accurate collection methods. So your estimation is only as good as your evidence. And if your major bits of evidence, in this case insects, are missed during collection, it will affect your time of death determination. So if somebody is out there and they're not trained in forensic entomology, or this is their first time doing it or something, and you don't collect the right insects, in this case we're looking for the oldest ones. Those are going to be the ones that show up first, right? So the oldest ones were probably the ones that were there first. If somebody misses those oldest ones, be it third instar maggots or pupa or pupal casings or newly emerged adults, whatever, they don't give those to you, then your estimation is going to be off. It's only as good as your collected insects. 3, you need to have good knowledge of perimortem activities. So really accurate PMI estimations depend on knowing what happened right around the time of death. Was the animal in distress? Was it injured? Was it sick? Was it wrapped or otherwise hidden? Was it buried? Since it is near impossible to know any of this information beyond a shadow of a doubt, that limits how much the postmortem interval is able to be estimated. All right. So if you find an animal just out and about, you have no idea if it was sick, if it had myiasis, if there were already maggots on that body before it died. In that case, what was going on with that animal? So you have to estimate PMI with that in mind. And whenever you give a presentation about postmortem interval, whenever you talk about it, that is one of the most important things in my mind. You have to be able to say, okay, given the information I have now, the insects on this body, this is my estimation. Now, this could change in the future if somebody tells me, oh, by the way, that animal is wrapped and frozen for 7 days or whatever, then that is going to change your postmortem interval estimation. But until you have that sort of information, you can only go off of what those insects tell you, what that evidence tells you. Okay. So those are the 3 major limitations for using faunistic studies. So it's these reasons that when we use those zoological methods, these faunistic studies of PMI estimation, we have to be careful when we talk about what we're actually estimating. Okay? Using insects, we are estimating the time from when the adult female found a corpse and oviposited on it to when that corpse was found. Let me say that again. When we are estimating postmortem interval, quote unquote postmortem interval, using insects, what we're actually estimating is the time from when the female fly found that animal, oviposited, and then when we found that animal. Okay? So, we are not necessarily estimating the entire postmortem interval. And this is something I talked about way back in Unit 1 and 2, about the time of colonization versus the time of death and postmortem interval. But that is what we are looking at. So the insect activity can give us clues, but little real information as to what happened before or around death, before those insects showed up. So we can glean what happened. We can get some clues as to what happened and then make those logical leaps, but we can't say beyond a shadow of a doubt this is actually what happened. So this is a little pet peeve of mine, I guess. And I think it's a pet peeve of a good portion of the forensic entomological community. When you hear people sit in court, or when you hear people on TV, or when you hear people talk about this and they say, this person died or this animal died 3 weeks ago at this time, 3 o'clock on Sunday. And you think, okay, really? There's all these limitations. There's all this stuff. Really, all you know is that the flies laid their eggs at a particular day. And now you know how old those maggots are. Sometimes that does match up with that postmortem interval. Sometimes it doesn't. Because like I just said, there are times when an animal was wrapped up and kept in a freezer for 6 months and then dumped out there. As that animal thaws, flies are going to fly in that animal. They're going to lay their eggs, and then you're going to get maggots, say, 2 weeks later. If you go up and say, this animal died 2 weeks ago, you are discounting any type of possibility that the animal could have been hidden, could have been kept from insect activity for weeks at a time or months. So it's this reason that we use that term time of colonization. We use that term to communicate these limitations. And so we are trying to let the world or let whomever know that we're actually estimating how long the insects have been on the body, not necessarily how long that body has been dead. Okay. So this time of colonization can easily be equivalent to the postmortem interval. Easily. I mean, We know that insects show up on a body within 15 seconds of death, or they can. There have been numerous studies that have tried to figure out when do female flies find a dead animal. So they've done a lot of work where they have sacrificed animals, they've euthanized animals at a scene, and then watched to see when flies showed up. And flies will be there, split second. They're just right there. They're waiting, ready to go. And there they are, laying eggs. Amazing. So they can be equivalent to that postmortem interval. But unless there is evidence supporting the fact that insects did show up right away, it should not be asserted. So you shouldn't say that insects showed up right away or that this is equal to the postmortem interval unless you actually know. Okay, so you have to acknowledge the limitations of the science. This is why way back I talked about looking at the confidence intervals, looking at all that sort of stuff. This is why. We have to be able to say we are 95% confident, or this is the range, or whatever. By using that language, you are acknowledging that there are limitations. So I like to use the language, this body was available for colonization on this date. So that way, in your report, in your speaking, in your testifying, you're actually acknowledging limitations of forensic entomology, yet saying exactly what the evidence is telling you. So you'll see in all of my reports, and in a couple weeks, maybe I think it's next week, I'll start talking about writing up reports, entomological reports. And so I'll say a lot of body was available for colonization. Bam. All right. Throughout the semester, I've peppered the lectures with some terms about insects on bodies. Now I'm going to define them for you now according to to their ecological categories. So we have all of these different ecological categories. And this first one is called saprophagus or saprophagus, depending on who you talk to. I always learned it as it rhymes with sarcophagus. So saprophagus, to say it slowly. Necrophagus and sarcosaprophagus insects. All of these type of insects are organisms that are those that feed on decaying matter. Okay? So, wow, I'm having a hard time saying these. Necrophagus in organisms are those that feed on corpses or carrion, while sarcosaprophagus insects are those that feed specifically on decaying flesh. Okay? So things that feed on corpses or carrion versus things that feed specifically on decaying meat. Now, these terms tend to be used interchangeably, although they have those slightly different connotations. But no matter the connotation, these ecological groups feed directly on the carrion itself. So it's these saprophagus groups that are most important when determining the time of colonization and therefore the postmortem interval. All right, so those are the main ones. We look at those, and those are the maggots, those are the flies, those are the beetles. Those are all those things that we spent the last couple of weeks looking at and trying to identify in depth. Next, predators. Predators are organisms that feed on other living organisms. So that is the big key. They're living organisms, and they consume the full prey item, killing it in the process. So a predator feeds on whole other living organisms, consumes that entire living organism, and kills it in the process. Okay, so on carrion, predators will feed on the necrophagus insects. Wow, on those carrion-feeding insects. And they are things like beetles, maggots. Some maggots are predators as well as carrion feeders, wasps, ants, those sorts of things. So you see, I've got a picture here of an ant or two ants feeding on a maggot. We talked about the Chrysomyia rufosis maggots that are facultative predators. So they feed on other living organisms and consume that whole organism, killing it in the process. Parasites. Parasites are slightly different. Parasites are organisms that feed on other living organisms, but they do not consume the full prey item. And most of the time, they do not completely kill it in the process. So most parasites will leave the organism alive after it leaves. There are some that kill it near the end, but they only feed on the living organism and do not consume the full prey item. So that's the difference between predators and parasites. Okay, so there's this picture here of a parasitoid wasp. So this is a wasp that will lay its eggs here in the pupa. Now, a parasitoid, that is an organism that is a parasite only in one portion of its life cycle. So in this case, this wasp is parasitic in its larval stage, but not parasitic in its adult stage. So it's called a parasitoid. We see those a lot in insects. Parasitoids are used a lot for biological control of other organisms. Now, we see two different types of parasitic organisms on carrion. There are those that feed on the living animal before it died, and those that feed on the necrophagous insects on the dead animal. So, animals associated with parasites tend to leave very quickly after the animal dies. So think of some of those parasites we've looked at. We've looked at fleas and lice. I think those are the two major parasitic organisms. Sand fleas, mites. All right, so all of those organisms that are parasitic on a living animal will leave very quickly. Now, there's no quantification of exactly how long it takes them to leave. Some observations show that they leave as algor mortis sets in, so as the body starts to cool down and the blood stops flowing. Others say they take a little bit longer. I've personally seen fleas leave the warm body of a pig that has been dead for about 20 minutes. But that has just been a couple of observations. I haven't quantified it. I haven't done any major experiments on it. So this can vary. Now, to me, this is a pretty useful bit of information, and it needs to be studied a little bit more in depth. However, there are some inherent problems with trying to do this type of experiment, so I think that's one of the reasons it hasn't been studied. Now, there are many parasitic wasps that prey on maggots and pupa of flies, and there have been a few studies on the usefulness of these organisms as time of colonization indicators. So since the parasitic development time is known, you could add on an additional layer of information as to how long insect activity has been in the area. If you say find some parasitized maggots or some parasitized pupa, you could say, okay, we need to add on not only how long those pupa have been there, but how long it would take that parasitic organism to get to its level of development based on its known life cycle. So there's a bit of information that you could add on using these parasitoids in order to add on to your time of colonization estimation. All right, so that's parasites. Now, omnivores. Omnivores feed on both carrion and the inhabitants of carrion. So both things. So there are beetles, there are some wasps, ants, and some maggots that are not only necrophagous insects, but they're also predators or something like that. So we call those guys omnivores. So officially, those facultative predators are omnivores. All right. Now, adventive species are those that use carrion simply as an extension of their environment. So we look at these as, those general soil-dwelling organisms, things like spiders, some of those ground beetles, all those type of insects that can live on or under the carrion, but will just as successfully live on or under a rock or a log or a bunch of leaves. So they're not attracted to the body specifically because it's a body. They're attracted to this dead animal because it happens to be in its area and it's offering some shelter, or maybe there's some moisture or something like that. So the insect was already in the area. It just happens to be under the body now. I get a lot of these types of insects sent to me from investigators who aren't trained in forensic entomology in any way. So I tend to get a lot of, centipedes or millipedes, those sow bugs, those pill bugs. Oh man, they're crustaceans. They like living under things. Those end up on bodies that have been there for a little while. I get a bunch of those. Like, what do these mean? It means they like something dark and moist. All right. Now, finally, accidental species. These are organisms that are not attracted to the carrion in any way, but end up there somehow. So, Sometimes they are seeking moisture, and there happens to be fluid around the body. Remember, the body will start to purge. Fluids, and so these insects will show up on these where this fluid is at. Others are just looking for a resting place. Maybe it was a butterfly flying by and it needed a resting place, and here's this body in the middle of a field. Okay, it's going to stop here. Just accidentally there. Others just happen to be wandering through the area and walk across or under the corpse. Centipedes and millipedes a lot of the time. This covers a wide variety of insects, so everything from butterflies to plant-feeding insects like aphids and grasshoppers and dragonflies. So I've seen aphids on a body. Aphids don't feed on bodies in any way, shape, or form that we know of. They feed on plants. But you find a body in a big field where there's a lot of aphids. Some of those aphids can be knocked off those plants onto that body. They don't tell us anything other than they were knocked off the plants. Dragonflies, same thing. I have used dragonflies. I think I mentioned this way back in the very beginning when I first showed you medical criminal entomology. I worked that case where I did collect a dragonfly from a car, and it was considered an accidental species. It was not attracted to that car, wasn't attracted. It just happened to be able to get into that car. And what it told me was that the car windows were open, so it allowed for free access. But that was the reason I collected it. I said, okay, this allows for free access for these flies. So this accidental species helped in that case. But other than that, it had no real bearing on the case. So they're all over the place. So these guys are not useful in determining postmortem interval, but they may be used to determine the circumstances surrounding death, like that dragonfly.
All right, so that's it for now. Let me know if you have any questions.